I am Jazzy, and I am studying nursing, so I just am starting the program this semester for the BSN and just finished my prerequisite courses. I don't know how to categorize myself because I'm a non-traditional student. I've done tons of years of school and have finally just dialed in this field. So yeah, going on to be a nurse.
I'm Quinn. I'm in the surgical technology program. I also am a non-traditional student, so it's kind of hard to also gauge exactly what year. I am hoping to go to the PA program when they open a PA program here. But yeah, I am about to start my second semester of the surgical technology program.
Histology is the study of tissues, and that is referring to the tissue types that exist in our bodies. So what we do is we look at those human tissues, for example, skin or liver or pretty much anything in the body, and we look at those under a microscope to view the normal and abnormal microscopic cell structures. Do you want to try?
Yeah, histology, like you said, is the study of tissues. To go off of what you were saying at the beginning, we do have different tissue types, and there can be a lot of different tissue types in just one sample. When you take anatomy and physiology, that's actually like the first thing that you learn is histology, I think. Mm-hmm. You know, after learning the anatomical regions and stuff, it's like you need to understand tissues. And a lot of times I think when you're in school, you learn something and then you leave it, and that's something that you learn it and you're using it over and over and over again through A&P 1 and 2. So it's kind of cool to actually be working with tissues and applying that knowledge towards pathologies, kind of like what you were talking about. So seeing differences, every body is different. Some people might have different illnesses or diseases or degeneration. So it's kind of cool to be able to apply what we're learning.
Yeah. I would say also that anatomy and physiology are the study of form and function. So anatomy being the form, how things look, what their typical structure is, physiology being how those structures function to perform the tasks that we need them to perform. So histology is the very microscopic understanding of the form and function of our body and the way it all works together. So our process has been just taking some of our cadaver tissues and processing them to view those structures on a microscopic level. So I would say as students, a lot of the histology is used just for learning, just for understanding the different organ systems, how they work. You know, when you look at skin, you see all the layers of the skin, all the different cell types within those layers. And so as a student, histology is really helpful just for understanding the body and how it functions. When you move beyond school, it's normally used as in pathology. So pathologists in a hospital will do biopsies on several different things. So I would say beyond school, histology is used more as a diagnostic tool to find pathologies and treat certain diseases.
Yeah, so if you think about if you were at the doctor and you had a skin biopsy done on a strange mole, for example, they would send that potentially to a histologist within the hospital, and they would basically check for abnormal cells. Well, personally, I feel really grateful to have some of the best instructors I've had in school, in all the schools I've gone to. Lucas, Witcher, and Kinsley are awesome professors. They make it really easy to be passionate about what you're learning about. And Lucas actually asked me if I would be interested in involving myself in this histology project that you actually started. And I was really excited about it because I really like the opportunity to learn more and to dive deeper. So that's kind of how I got started. It's been really fun actually so far. Yes, I did start in anatomy and physiology in their classes. I did take anatomy and physiology 1 and 2, and then I started prosecting. I really applied myself. I've shown up to all the extracurriculars whenever they have a surgeon come into the cadaver lab to do a surgery. I'm there. And I don't know if it's that type of like excitement and interest that he was like, you want to do this? But that's what got me going, and it's been really fun. So
yeah, so as anatomy and physiology students at the Missoula College, we all get the opportunity to look at the cadavers in the cadaver lab, which is very unique, I would say, especially at a 2-year college. So I was in anatomy and physiology 1 and 2, and the instructors here have made that lab, which can be a very intimidating space for many, so inviting and so welcoming and such a unique area to learn, because we have all these great models, so many great models and so many great texts and online resources, but the opportunity to actually look at the human body and to be able to see how it's layered and see the sizes and the positioning of all the organs and everything is just super unique. So that is definitely where the flame began, is in the cadaver lab, just as a student with the opportunity to learn in there. There are a select group of students, select only because it is limited in numbers, but some students who are interested get the opportunity to be a prosector in the cadaver lab, which is similar to dissection, but instead of just dissecting for learning, you are dissecting to preserve and display those tissues for other students. So Quinn has prosected, and I have prosected as well. And it is from there, as a prosector, that Lucas kind of threw out the idea of different projects that we could work on. And one semester, I think it was my second semester as a prosector, he mentioned that he wanted to see if histology could be an option, if our cadaver tissues could be processed for histology. And it was as simple as that, and it sounded very exciting to me because histology is such a foundational understanding for pretty much all of the health sciences, and it's lab work. You know, you get to work in a lab and you get to use your hands, and that sounded really exciting to me. So he kind of threw out that idea as an option of a project to work on, and I was like, oh, that sounds awesome. Like, I want to do that. Let's figure out how to do that. And from there, it was very much a process of making connections with folks who had experience in that area, and many hours just spent online just trying to find what I could. Not a ton of resources for specifically cadaver tissue being processed for histology, so in some ways it felt like we were kind of pioneering this project. But it started as an idea planted by Lucas, and then I kind of just ran with it and was super grateful when I learned that we have a histology lab on the main campus. So I connected with those folks, and they really held my hand in those first months as I was figuring out how to undertake this big process. So yeah, started as a seed planted and really was just presented as an opportunity for me to deepen my learning, to do some hands-on research, and to kind of like take the reins a little bit and do some learning and have access to equipment and things that really, kind of helped me get there. So yeah.
Well, on a personal level, I think working on this project has made an opportunity for me to expand my understanding and my learning. You know, I'm going into the healthcare field. I want to be as well-rounded as I can be. and informed. And like you were saying, it's foundational what we're learning about. On a bigger level outside of myself, being able to take our own cadaver tissue and provide something for other students to look at, even having microscopes and slides available, I think that really improves the kind of just like the aspect of learning, you know, having something hands-on, having something that's ours, having something also to compare. You know, we can take one cadaver and we can look at heart tissue, we can take another cadaver and look at heart tissue, and you can see how different people are on a cellular level, which I think is really powerful. It is a big difference looking at something of your own, something that you can hold in your hand versus something you're looking up on the computer, which is what we've been doing. So I think this is really going to also improve the access and the education that students, future students, are going to have, which I find actually really exciting.
Yeah. Well, I would say that this is exciting to me on a personal level because of what we keep saying. It's a foundational study to all health sciences. Understanding, like that's why we start with anatomy and physiology, because you have to understand the basics of the human body before you can go much further than that. And histology is the literal basics down to the cells performing those functions. And to me, that's just so interesting that there's this whole world that we can't really see with the naked eye that we get to unlock through the process of creating these histological slides. So to me, it's very just exciting. It's very fun and foundational, and I think in some ways, personally, it really has helped me to make connections with really brilliant professors and even folks in the medical community in Missoula. I've been able to make connections with pathologists and mostly pathologists and some students studying histology. So it's been cool, just kind of almost as a personal development in many ways, alongside all of the learning that has come on the topic of anatomy and physiology. Being able to connect the entirety of an individual that you're seeing with those microscopic cell structures that we are creating is really cool. So when we talk about different like pathologies, we have one cadaver with a very large heart, and that's something you can actually see on the histology slides that we've created, that those cells are enlarged. So just things like that, being able to look at the cadaver that we've studied all semester and make connections with some of the pathologies or things that we're seeing is really cool. I think that the skills on a microscope are awesome, and it's not something that even either of us did in our time in the anatomy and physiology program. So to be able to provide those slides for students to actually look at the slide and be able to navigate around what they're seeing and see a structure that's nearby that they might not have seen had they just looked at the online resources with, you know, this perfect epithelial, you know, sorry, it's been a while, but this perfect structure that has just been selected for them. Instead, they get to look at the whole slide and kind of understand the variations in what they're seeing. And I think just in general, whenever you can make learning hands-on, goes a long way and just is retained so much better for most folks.
Well, I do think that we're bringing, like we mentioned before, we're bringing something to the classroom that makes learning more hands-on, more accessible. There's more diversity in what you're seeing as well. If we're just looking at slides online, it could be one kind of portion of an organ that you're looking at, like the tissues themselves, but we have access to a more full encompassing picture, if that makes sense. I do think with processing cadaver tissues, there's a whole host of uncontrolled variables that we've come in contact with, you know, such as preservation times, how does certain preservation or fixatives like affect the tissues long term? You know, are the tissues breaking down before you have access to working with the cadaver itself? There's a lot of factors that are and variables that are way harder to control than if you were to just take a biopsy and take it right to the lab if you were in a hospital. And so it's definitely a learning curve. So I also think in that way it's both a challenge and also a really good way for us to learn how to differentiate, oh, this is a processing error because we're using cadaver tissue and these variables can't be controlled, versus, oh, this is a pathology, there's something different here that we're working with. So I find that to kind of be like the next step for me as we continue this project, is to kind of identify that a little bit more, start learning about the pathologies that we're looking at, connect more with the community. I think as we grow and as our learning on this grows, we are going to be able to offer more insight and a well-rounded kind of approach and understanding of what we're working with. And I think that is really unique, especially if we can build some sort of manual kind of to be like, hey, this is what we're doing, pass it on to the next people who want to take this on, who want to work with. Maybe we get a new cadaver, maybe there's something different, and they have a blueprint to work with, because like Jazzy said, there's not a lot on the internet about processing histology from like cadaver tissue. It's not really accessible online. There's not a lot on there, and so maybe we'll be creating a resource for future students that can be really positively impactful.
Yeah, I think definitely what we're doing is unique. Like Quinn mentioned, at the start of this project, Lucas and I found about 2 articles that talked about creating histological slides specifically from cadaver tissues. And the aim of these papers was basically to figure out whether or not these tissues were viable to be processed for histology, and whether there is an opportunity to learn in this way. And so it's really, as far as we know, not something that's done very often. And the part of why that's so cool is because it's a really beautiful thing that these people have donated their bodies for our learning, and creating these slides is a way for us to preserve the legacy of their donation, which is kind of how it ties into the legacy project. Just being able to, you know, this heart that we have with these enlarged cells, we now have a viewing of that pretty much forever. You know, these slides will last for a very long time. So it's unique in that people aren't really processing cadaver tissues, as far as we know, for histology. And it's a great skill for us to be able to learn, like Quinn said, how to process those tissues. A lot of trial and error. Difficult to get into the steps, but it's about, let's see, probably like 7 steps that are quite time consuming, that we have to really dial in and figure out what produces the best results. So definitely one of our goals is to create steps 1 through whatever of how to get the tissue from the cadavers in the lab onto a slide successfully. And yeah, as far as we know, it's not being done too much. So
I think it would be really cool in the future if we are able to have basically an archive, you know, if we continue to pass this down and in its own way have our own kind of legacy of this histology project, and we have future students doing what we're doing and we create a blueprint, you can have an archive of, you know, it's like you can have a folder of, this is all heart tissue, and of all these different cadavers, this is a different part, you know, of the heart. And I think that's really cool to be able to compare, to learn, have a resource for so many students, and have that, like you said, it's pretty much forever. You know, when we take pictures of these slides, we can, it's really cool to think about being able to, immortalize these things for for students. I don't know.
And the variety, there was another word you used that's it's leaving me right now.
I don't know.
But yeah, the variety of viewings is really cool because
diversity.
The diversity. Thank you. A lot of our learning of histology has been on Histology Guide, which is an online resource that's awesome. You can search by tissue type or organ, but like Quinn was saying, sometimes you might look at the connective tissue and you have this exact slide memorized, but it really could look so many different ways. So to be able to have the diversity of viewings, I think it just creates a more holistic understanding, as well as seeing the cadaver, seeing their tissue under a microscope. All of these things just kind of give students the opportunity to view things from many different angles and just have a more wholesome understanding.
Well, I actually just applied to a conference for STEM, both professional students, kind of that whole realm. It's to help connect different people in the STEM world, create more just opportunities, and also just open kind of doors for a lot of people, which I'm excited for. But they're also helping fund trips to conferences, and I would really like to work on an abstract or a manuscript on what we're working on and present that. I know Lucas really wants this as well, but present at the Human Anatomy and Physiology Society conference next spring. I also, with this project, like I was mentioning earlier, I'd really like to kind of dive into pathologies, kind of get into the nitty-gritty now that we have a lot of different samples, and also kind of fine-tune. There's some tissues that are not as easy to process that we haven't nailed down, and potentially kind of fine-tune our processing as well and create. I know we didn't get into this too much, but it would be really cool on the machines that we use in the histology lab to create our own kind of, what are they? They have these like settings that you can create yourself. So you are fine-tuning all of the composition, and it would be really cool to have our own presets for our machines that we use in the histology lab based off of our research.
Yeah. I think for me, one thing that's really important since I started this project is, like we've mentioned, just creating a way to pass that down to future students so that they can have that experience in the histology lab, creating the slides, and that we can continue to just add to the archive of tissues by organ and tissue type and pathologies, and just have this amazing resource for students here to have a more diversified learning experience. So that's one thing that's really important to me, is to just have something to pass along to the next student. And then I'm actually also hoping to do some shadowing in some pathology labs in town. I am going into nursing and still open about my specific area of interest, but I find this study very exciting, and so continuing to just make connections around town. And like with most of my schooling, I feel very open and kind of learning as much as I can and following the paths of excitement and passion, and maybe this will be one of them. I mean, no, probably. Yeah, I actually studied some of these things 12 years ago maybe, and even looking at the cadavers felt like a cool opportunity, and looking at histology was exciting, but to be able to have our hands really in this so much is, not something I thought I'd be able to do, and has been so cool. So cool, yeah.
I agree. I feel the same way. I've always been interested in healthcare-related fields of study. But I'm happy that I took time off of school, and I feel really confident about what I want to do, because I've been way more just like go get it. towards my education. And I think when you are willing and ready and open to your learning and to the opportunities that pop up all the time, more opportunities just come your way. You know, you'll start to get emails that are like, oh, we have this visitor coming to the school, and if you, it's wild how much opportunity, you know, and how many doors are open when you choose to participate in those things. So I have also been kind of shocked at what I've been able to do here and surprised, and it's been super lovely and wonderful. But yeah, I don't think I would have ever expected to be doing this much, and so it's really exciting and it's really fun. Yeah.
Tons. There's been a lot of failure, I would say, like before Quinn came along, just personally, but then even since you've been here, it's been a huge learning curve.
Yeah.
Like, I don't know if you've had anything like weird. What do you think?
When I think about the moments that feel the most blooper-ish, it's like always when we're in the histology lab and something's going awry. The microtome, when there was this moment where it was we were like, what is wrong? It's not cutting, but it was just, it's hard to describe the machine.
Yeah.
But we were just like really struggling with it for a while, and we were like, oh, it was kind of a simple, the blade was just all the way forward, so it couldn't continue to come out to cut the slide, right? To cut the wax block. And so we're like, I don't understand what's going on. And then it was just, oh, we need to push it back.
Just a lot of, the equipment. It's like pretty high tech, you know?
Yeah.
And a lot of small parts that have to function in a very precise way.
Yeah.
But yeah, I don't know. The most fun part is definitely the sectioning, which is the step where you take the tissue that's been dehydrated and infiltrated with paraffin wax, and it's set into a block of paraffin wax. You take that block and you create very thin slices that get mounted on the slide. And that is probably the most time-consuming, definitely most involved, most hands-on, funniest.
You can't breathe.
Maybe, you have to cut and be like move the tissue to the side, because if you even breathe out a little, it's going to fly away.
Yeah.
The thickness is 5 microns, and I can't explain to you how thin that is, but it's real thin.
It's like if you took a sheet of paper and sliced it a bunch of times and made it through the flat, part of it, and it's just a wisp,
Yeah.
You'll finally get a perfect little cut, and then you'll try to move it, and it just flies away, and it's heartbreaking.
Yeah.
But it's fine.